So now in this video we are going to continue with our discussion on the lens of the human eye. In the previous video we talked about the anatomy of the lens, the different layers of the lens, and the anatomy of the different structures associated with the lens. In this hopefully short video we'll be talking about a few small topics related to the lens. The first one, as you can already see from the heading that is given over here, is the antioxidant mechanisms that are present in the lens. Now, what does this mean? See, the word antioxidant, we know that our body has, or rather requires, a lot of antioxidants in many I would say different bodily functions. We often have, I would say, heard, or rather we have often seen advertisements on televisions regarding some good healthy product having a lot of antioxidants and stuff. So the thing is, what exactly is antioxidant? So it is given in the name itself. Anti means against and oxidant means oxygen. So the lens, this is something that I forgot to mention in my previous video, but I'm going to mention it here because it pertains more to this video. Lens somehow has a very low oxygen content environment. Okay. Now this very low oxygen content is very necessary. See, why am I saying low oxygen content? See, lens, at the end of the day, what is the lens composed of? We already saw to this in the previous video, right? If I can go back to my previous video and show you the different layers, then you will see that the lens is at the end made up of different layers, and the layers are made up of mostly different cells, right? So if they are cells and if they are living cells, then obviously they'll have what? Requirements. They'll have requirements for oxygen, because they're living cells, so they'll need to carry out their functions, and for that they will need oxygen, they'll need glucose, they'll need metabolism, and all of that is what we're going to talk in this video. So long story short, they need oxygen, but the oxygen content in the lens is low. Why is that? That it has been found that high oxygen content in the lens, if for some reason there is high oxygen content, it is actually involved with, or rather it is actually linked to high cataract incidence. Now, what exactly is cataract? We'll talk about cataract in a very big detail, very detailed way. We'll talk about cataract, but for now just know that cataract is the opacification of lens or any of its associated structure. Okay, so lens or not associated structures, I would say lens or its capsule. If there is opacification of the lens or its capsule, then we call that to be cataract. And obviously you would all have heard about cataract, right? Cataract is so common. Anyways, so high oxygen content has often been found to be related to high cataract incidences. So we do not want that. We want the lens and its surrounding medium to have low oxygen content. To maintain this, there are different substrates, or rather there are different enzymes, there are different products, or rather substances, chemicals you might say even, which actually help in lowering the oxygen content. They are known as antioxidants. What are these antioxidants? The first one is something we call to be an enzyme called superoxide dismutase. Now, what exactly is the function of superoxide dismutase and how it is related to being an antioxidant, or rather a very potent antioxidant in our body? All of this will take up a long time, so I'm not going to explain it here. Rather, in a subsequent biochemistry video, I'll talk about this. Okay, there is a huge topic in biochemistry related to antioxidants, and there we'll talk about superoxide dismutase. Anyways, now the second one, another enzyme called catalase. Thirdly, something known as very famous enzyme, glutathione reductase. All of these will come across when we'll talk about in detail metabolism in biochemistry. Okay, so these are all related to biochemistry. Obviously, this is a biochemical topic at its core, right? We're talking about antioxidant mechanisms. So obviously this is a biochemistry topic. Anyways, so these are three enzymes which are very much related to antioxidant mechanism in the lens. Others include vitamin C, vitamin E. Now, there is something to be noted here. Originally, vitamin A is also an antioxidant, but it is not an antioxidant in the lens. So even though vitamin A is an antioxidant in general, but not in lens. In lens, it is vitamin C and vitamin E. Okay. Another interesting mechanism, and this is very, very interesting. A very interesting mechanism by which low oxygen content is maintained around the lens is, if I remind you what exactly is present around the lens. See, if I go back to our previous videos, or rather previous slides rather, you will see that behind the lens there is this thing called the vitreous chamber, right? The entire posterior segment is present behind the lens, which is composed of the vitreous. Now, lens, to bathe the lens in a low oxygen content, its surroundings should also be low oxygen containing. So for that, a very interesting thing is that the vitreous chamber, that also has very low oxygen content. The applied aspect of this point, this last point, is that sometimes, and we will see when, we have to cut out the vitreous. We have to extract the vitreous. We have to make a hole in the vitreous. That procedure is known as vitrectomy. Vitrectomy means that you are taking out the vitreous, you are making some kind of a change in the vitreous. So if you make this kind of a change in the vitreous, if you take out the vitreous or something like that, what is going to happen? You are basically disturbing the low oxygen environment that has been created by the vitreous for the lens. And hence, it has often been found that procedures or other conditions which need vitrectomy, after vitrectomy, often there is increased chances, increased incidence of cataract. Cataract means opacification of the lens or its capsule. Okay. So this is, in short, a very small topic, the antioxidant mechanisms in the lens. So what is going on? Just a quick recap. The lens needs low oxygen. Remember this point. After dealing with just another small topic, we will go into the proper metabolism of the lens. When I talk about the proper metabolism of the lens in this video, this statement of the lens needing very low oxygen, or the lens requiring a very low oxygen content, this statement is going to be very handy when we'll talk about the proper metabolism of the lens. So do make a mental note of this statement. Now, just one small topic before we delve into the full metabolism of the lens. That is lens paradox. What exactly is the lens paradox? Very simple. As one ages, during old age, what happens? There are two different changes that are going on in the lens of a person. What are these two changes? First of all, if this was the lens of a young person, if you compare the lens of an old person, you will see that the lens has become somewhat like this. What is this? This means that there is an increase in the curvature of the lens. Actually, what happens is the lens with age becomes more and more rigid and thus becomes more and more curved. Okay. Now, if the lens becomes more and more curved, from our previous video discussion, I have done a very, very detailed discussion on this. If the curvature of the lens increases, what is going to happen to its converging power? Yes, it is going to increase. So that means that this will lead to increased convergence. Okay. That means that now the light rays traveling from infinity, they will be converged much in front of the point compared to where they were being converged when the lens was flatter. Okay. So the converging power of the lens will increase with increase in its curvature. As simple as that. But there is something else happening in old age as well. The next thing that is happening in old age is that the refractive index of the lens is decreasing. So if the refractive index, what exactly is the refractive index? The refractive index is of anything is how much it can alter the trajectory of a light ray when the light ray is traveling from one medium to that medium. So for example, I'm saying that we use the Greek letter mu to denote refractive index. We all know this. This is all basic physics. So refractive index of glass is basically how much glass is able to alter the path of light. So if this was air and this is glass, this is the normal, this is light ray, then the light ray will move towards the normal, right? This is called refraction. Okay, this is refraction, and this is moving towards the normal because glass is optically denser than air. This is all pure physics, so we are not going into that. So basically what is happening, what I'm trying to say here is that if the refractive index of something is decreasing, that means the power of that substance, the power of that material is decreasing. So that means there is decreased convergence. Decreased convergence. Power obviously here means optical power. Okay. So and this is I and this is R, and mu, if I remember correctly, is sin I by sin R. Let's not go into all that. Okay, let's not go into all that. So anyways, I just remembered it and I just blurted it out. Now look carefully. One point is leading to decreased convergence, one point is leading to increased convergence. What happens in old age many times is they balance each other. They balance each other. This is known as the lens paradox. This phenomena is called the lens paradox. Okay, as simple as that. This was a small topic, nothing much to really gain from here. Okay. Now let's come to the real topic at hand, the metabolism in lens. Metabolism in lens. What exactly is metabolism? Metabolism means metabolizing. Metabolism means for requiring energy, or rather I need energy, so I will use some energy source for doing my day-to-day functions. I will break down that energy source into some usable form and use that for carrying out my day-to-day functions. This in short is metabolism. I know this was not short, but this is in a very layman term what metabolism means. So basically we are breathing in air, we are eating food. This is all very basic elementary things that I'm talking about. Okay, obviously all of you, if you're watching this video, this is a very niche video, right? I'm not talking about just the human eye here. I'm talking about the lens in the human eye and the metabolism of the lens. So this is a very niche topic. I don't think a lot of very science, not non-science people are going to be watching my video. So I'm going to take some liberties here, right? I'm going to assume that you are somewhat capable of understanding what I'm saying here. Okay, I'm going to assume that you have some basic level of understanding. So you all know what metabolism is. If you don't, then you should go back to biochemistry. You should watch biochemistry videos or some elementary videos on the metabolism. So having gotten all of that out my system, let's come back to this. So metabolism in lens. Why exactly does the lens need metabolism, by the way? It's very simple. I just said that lens is composed of what? Lens is composed of living cells. Yes, lens is composed of living cells. So if there are living cells anywhere in the body, they will need what? They will need energy. And I need to provide them energy. How will I provide them energy? What is the universal substrate? What is the universal, you might say, substrate is the word. For energy, they'll need glucose. So that is where metabolism comes into play. Okay. Now, Remember that point that I had made and told all of you to make a mental note of. I had said that lens is a low oxygen environment. That means that all of the respiration that will be going on in the lens will be anaerobic. Anaerobic. Rather, it would be a little stupid to say all. Rather, let's play it safe. Let's say greater than 80% is actually anaerobic respiration. Anaerobic means not requiring oxygen. And yes, it is very, very, I would say, pertaining. It is very, very apt, rather, that anaerobic respiration will go on in a low oxygen content, or rather low oxygen environment. When we exercise a lot, our muscles start building up lactic acid. Why is that? Lactic acidosis, why is that going on? Because of anaerobic respiration. Oxygen, there's not enough time for the muscles to utilize the entire aerobic glycolysis. Rather the aerobic cycle, not aerobic glycolysis, the aerobic Krebs cycle, all of that, no time. So let's go for the anaerobic glycolysis. That is why pyruvate, and then from pyruvate to lactic acid. All of this is basic biology. Now, so the first thing that we need to see is what exactly is the glucose in the lens metabolized by. So let's make very quickly, let's make a list. So glucose has entered the lens, been carried to it somehow. How, I'll just tell you later, or rather right now I'll tell you, but just in a few minutes. How are you going to deal with this? How is the glucose metabolized in lens? Okay, the glucose in lens, I have already told you one of those ways that the glucose is going to be metabolized, that is anaerobic glycolysis. In fact, this is the major way of metabolism of glucose in the lens. Greater than 80% of the glucose is metabolized by this way. Okay. Second is the aerobic way, that is the Krebs cycle. Krebs cycle, or even the HMP shunt, hexose monophosphate shunt. This pathway, this is more or less 15%. And the remaining 5%, or rather less than 5%, is by something called the sorbitol pathway. Okay, let me erase this and let me actually write this in a way that you are able to understand, because this is a word that you might not be very much familiar with. Sorbitol pathway. The sorbitol pathway is a very, very important pillar in our discussion on the metabolism in lens. So anaerobic glycolysis greater than 80%, Krebs cycle or HMP shunt less than 15%, or rather greater than 15%, you might even say, and less than 5% the sorbitol pathway. When exactly are these pathways functioning and what exactly is going on? Let's explore all of these in a little bit more detail, not like a lot of detail, because HMP shunt, Krebs cycle, glycolysis, these are all topics of biochemistry, right? Even the sorbitol pathway. So we are not going to deal with all of these in a lot more detail here. Rather, we will see the applied aspects of these. So first things first, as I was mentioning in my previous slide, where exactly does the glucose come from? Glucose in the lens, think about it. What exactly is the conduit, or rather what exactly is the transporter of different substances in our body? Think about it. What tissue, or rather what cells in our body transport different substances all throughout the body? There is an entire conducting system in our body, right? What exactly is the conducting system made up of? Arteries and vessels, right? Arteries and veins, rather, blood vessels, blood, lymph. All of these are the conductive system in our body. They conduct things, they carry oxygen, bring out carbon dioxide, blah, 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 all of that, metabolites and all of that, blah, blah, blah. But is lens a vascular structure? No. Lens is avascular. We know this. We already know this. lens is avascular. So that means blood cannot be the way that you transport glucose to the lens. What else is there around the lens? If I go back to one of my previous slides, then see in front of the lens over here, this is white, over here, this entire thing. What is this? This is the aqueous chamber. This is the aqueous chamber. This is actually the anterior chamber, and that contains aqueous. Aqueous is a watery fluid which is secreted by the non-pigmented epithelium of the ciliary body, right? We all know this. We have already talked about that. One of the functions of the aqueous, if you remember, was transportation of different metabolites. So it acts as the conduit or the way that glucose is transported to the lens. So basically, aqueous glucose, actually, where is it coming into the lens from? Glucose to lens, or rather aqueous to lens, there is diffusion of glucose. Okay, diffusion of glucose from aqueous to lens. So basically, suppose this is the lens, this is the ciliary body with the iris. In front over here, this is the aqueous. So from here to here, there is diffusion. Okay. Now, see, I had mentioned in my previous slide about these three mechanisms by which glucose is metabolized. So after the glucose came into the lens, now it will be metabolized. What is the primary way in which it is going to be metabolized? It is going to be metabolized by anaerobic glycolysis, mostly. Sometimes by Krebs cycle or HMP shunt, about 15%. But few times, very few select times, it is going to be following the sorbitol pathway. Now, what exactly is the sorbitol pathway? Let me quickly give you all an overview. So, suppose this is glycolysis going on. Glycolysis was anaerobic, right? Anaerobic glycolysis was going on. But suppose the glucose that is coming in at a constant rate from aqueous, for some reason, more and more glucose is coming into the lens. More and more glucose is coming into the lens. More than it normally should. That means that person has an excess of glucose in his or her body, which means that person is suffering from hyperglycemia. So, in case of hyperglycemia, that is, there is a lot of glucose, there is a lot of substrate for glycolysis, too much, then what will happen is this hyperglycemia, excess of glucose, will lead to rate limiting feedback inhibition. Rate limiting feedback inhibition of the glycolysis. Now, mind you, 80% or even more of the metabolism was through glycolysis. But due to hyperglycemia, now there is a rate limiting feedback inhibition of glycolysis. So, no longer will glycolysis take place. So, the other pathways will become more active. More active, actually even more than Krebs cycle, will be the pathway on sorbitol pathway. So, sorbitol pathway will become active. So, sorbitol pathway becomes active when there is hyperglycemia. Okay. Now hyperglycemia, what exactly can hyperglycemia, or rather what are the conditions where hyperglycemia can be seen? Obviously goes without saying, diabetes mellitus. But diabetes mellitus is more or less something that is concerned with the adults, less in children. We'll see that there is a variant of diabetes mellitus which is more in children, DM type 1. Anyways, we'll see that in medicine. So more in adults. What about in children? In children, it has been found, or rather, yeah, it is in children, more in infants even. You might even say infants. There is another inborn error of metabolism called galactosemia. Galactosemia, seen in children. So both these conditions will lead to a very high concentration of sugars in the body, leading to hyperglycemia. Hyperglycemia leads to rate limiting feedback inhibition of the anaerobic glycolysis that was going on, and leads to the activation of the alternate pathway called sorbitol pathway. What is going on in sorbitol pathway? Basically, normally sorbitol pathway would have been less than 5%. Now it will be more than 20%, even 30%, 40%, depending on the degree of hyperglycemia. What is going on in sorbitol pathway? Let's see very quickly. So what is our starting substrate? Glucose, right? Glucose, instead of going through the anaerobic glycolytic pathway and being converted into pyruvate, rather it is converted into something called sorbitol. And the enzyme which is needed for this is aldose reductase. We know that glucose is an aldose, so aldose reductase. Aldose reductase is the name of the enzyme. Okay, so glucose is reduced to sorbitol, and sorbitol, it is freely convertible to fructose. Okay, now what exactly is the applied aspect of this? Why are we studying all this in so much detail? What exactly is the meaning of this? If you become an ophthalmologist, what use does this serve? What use does knowing the sorbitol pathway serve you as a clinician? Now is the applied part. So listen very, very carefully. I had said, and I have actually spent quite some time explaining to you how glucose gets into the lens. Glucose gets into the lens by diffusion, as I explained in this image over here. Diffusion from the aqueous. In fact, glucose can diffuse in and out of the lens very freely. No hindrance, no obstruction, no problem. Everyone loves glucose. Grand time, amazing time. Lovely. Problem is this one, sorbitol. Sorbitol forms, or rather sorbitol creates a very, very potent problem, a very, very strong problem, dangerous problem, due to the fact that sorbitol cannot diffuse. It cannot diffuse in and out of the lens. So what will happen? The more the sorbitol pathway is active, the more sorbitol is created. More sorbitol is created, the more it accumulates. The more it accumulates. Right? So the more it accumulates, what's going to happen? Sorbitol at the end of the day, what is it? It is a sugar, right? And a sugar compared to water, what is it? It will be more in what will a sugar make water? Water will be made more concentrated by a sugar. What I mean to say is sorbitol will lead to increase in the osmotic pressure in lens. Osmotic pressure in lens will increase. Are you understanding what I'm trying to say? If a sugar It is not supposed to be there, right? If it had been able to diffuse in and out freely like glucose does, no problem. We would have no problem, because then it would not have been able to affect the concentration of the fluids within the lens or of the lens itself. But sorbitol, it is accumulating. And what is going to happen? It is going to make the environment more concentrated. So in a way, it is increasing the osmotic pressure. And if there is increased osmotic pressure, what will happen? Fluid, liquid, or rather fluids rather. Fluid is the apt term, not just liquid. Liquids and gases, so fluids. Fluids move from a region of their higher concentration to a region of their lower concentration, right? That is what diffusion or osmosis is. So increased osmotic pressure within the lens means that there is increased concentration of the sugar within the lens, which means there is decreased concentration of the fluids within the lens. So now there is more fluid outside the lens than there is inside the lens. So what will happen? Fluid is drawn in the lens. Fluid is drawn in the lens. And what will this lead to? This will lead to, if fluid keeps on being drawn inside the lens, this will lead to opacification of the lens. And what is the opacification of lens known as? I have already told you. This is known as cataract. Cataract formation. This is actually a special form of cataract known as sugar cataract. Okay? So that is why knowing the pathway and knowing the applied aspect of the pathway is so important. Okay? So this is mainly, I hope I was able to convey to you this entire pathway and the applied aspect of this. This is more or less the metabolism of lens, and this is more or less the applied aspect of the metabolism of lens. So hopefully, I'm trying to make this video short. Hopefully I was able to convey to you, I was able to deliver to you what I was trying to teach, convey, whatever. And hopefully you are able to understand this topic. So thank you for tuning in. And if you've made it this far, thank you very much. And we'll meet again in the next video where we'll talk about, hopefully, we'll talk about cataract. Thank you.